Hey, I'm going to go ahead and do the second video now while the ball game's going on. You'll watch this the day after anyway, so I'm not worried about it. But we're just going to continue where we left off. We got done talking about the journey that a signal takes as it travels through the neuron. We're going to continue that. We're going to get a little more detailed. The first thing we want to talk about is the myelin sheath. Again, it's sort of the insulation, I guess you could say, and it protects the axon and the electrical signal that it's carrying. Again, just like the orange plastic coating on an electrical cord. The more you have, the better the transmission is, and that's something to always know. Is that really, that's why I give you an example. We have an RV. If you've ever been past my house, I've got an old RV. Excuse me, and we will run an electrical cord to the house to run the air conditioner in it. Well, if I use one that's too small for the length, by that I mean if I use one that doesn't have the right big enough diameter or doesn't have enough insulation and it's really long, resistance builds up and actually it will get hot and it will actually trip the breaker. But if I use one that's really thick and that has real thick insulation, that electrical current moves a lot better. It doesn't quite have the resistance and it just works better. I don't blow any fuses or trip any breakers or anything like that. So, sorry, I just watched Georgia Tech run a big 40-yard play. Sorry. The myelin sheath. So that's what the myelin sheath does. It is a insulator. It's made up of what's called Schwann cells, which is a specific type of glial cells. They create the fatty substance that insulates. All right, now we're going to talk about this thing called action potential. And we'll talk a little bit more about it in class, but this is what moves the signal. And this is actually gets a little bit into physics and chemistry, and so I'll try to do as best I can. But as information enters the cell here at the dendrites and goes through the cell body, and the nucleus decides, yes, I want to send this. All right, it enters into the axon, which we've talked about, but we've not talked about how it moves. All right, and that's what the action potential is. It is the electrical charge. This is the fire signal of the neuron, and that causes the neurotransmitters to be released at the terminal buttons, which we've not talked about the neurotransmitters or the terminal buttons yet. We'll talk about that a little later. All right, but essentially what happens is the polarity changes between the inside and the outside of the axon. So you all have seen magnets where you can sort of move a magnet if you just turn it around. If you've got it, you'll have a positive side and a negative side, and you can just kind of push that. That's essentially what happens as it enters, or as the signal decides to get sent. The inside of the cell turns to a positive charge, outside turns to a negative, and it shoots it down through there. So, and that is called action potential. That is the electrical charge that sends that signal. The axon gets its energy from charged chemicals called ions. In its normal state, the ions have a small negative charge called resting potential, and that sort of this here shows it's negative on the outside of the cell. Well, when it's resting or when it is not firing, it is also negative on the inside. All right. When the cell decides to send that signal, the negative balance can be easily upset. However, when the cell becomes excited, when it decides to send that signal, it triggers the action potential, which reverses the charge and causes the electrical signal to race along the axon. So it will When the cell decides to do its thing, all right, this will switch from negative to positive and push the signal through. So this is just a short little quiz type thing. A brief electrical charge that travels down the axon is called what? And we'll talk about it tomorrow if you don't know. It should be obvious. It is action potential. All right, now we're going to talk about this thing called absolute threshold. And this is the amount of signal that it takes essentially for the cell to fire. You know, the neuron, it's a decision maker, all right, and that's what the nucleus does. It receives information from thousands of other neurons. Some are excitatory, like pushing the gas pedal, or others are inhibitory, or putting on the brakes. If the excitatory signals minus the inhibitory signals exceed a minimum intensity called the absolute threshold, then action potential is realized. Essentially, think of it like this, okay? I'm going to try and back you up a little bit. You've got all these cells sort of giving all of this information. All right, these are the excitatory. They're trying to excite the cell. Then you have other cells or other neurons that are sending stuff that is inhibitory, so of pushing it down. All right, so you've got these two things sort of working against each other. All right, well, imagine that, let's say my eye level right here is the absolute threshold. What that means is if the excitatory signals overcome, or there are more excitatory than inhibitory, all right, then it raises it up. Once it gets past my eyeball, boom, then the action potential is realized. It sends it. Another way to think about that is the lever on a toilet, probably one you got at home, where you push the lever down, and it lifts up that valve, and the water from the tank goes into the bowl, and everything that's in the bowl goes down the drain. That's an example of absolute threshold, because as you're pushing down on that lever, if you push down a little bit, then nothing happens. Push down a little more, but at some certain point, all right, that lever eventually raises. All right, that is an example of the absolute threshold. Once it raises, then it's done. All right, then everything happens. Same thing in a cell. Excitatory signals, inhibitory signals, and they're always sort of in constant battle. All right, once it hits that absolute threshold right here, all right, then it's shot. Then the cell fires. So that is absolute threshold. And here's an example of how that works. You've got the information coming into the dendrites, cell wall or cell body. Nucleus says yes, we're going to send it, and as this is how that works, they switch, and that pushes it through. And there is a principle called all or nothing. It's kind of like shooting a gun. You don't, or even flushing a toilet. All right, you don't. Most times you can't halfway flush a toilet, or you can't halfway fire a gun. The gun either goes off or it doesn't. The toilet either flushes or it doesn't. All right, that is the all or nothing principle. Once it hits that absolute threshold, boom, it fires. All right, it doesn't half fire if it gets to almost there, or if it gets just a little bit past, it doesn't half fire. No, it either fires or it doesn't. And again, think about, my dog is sleeping over here. It's snoring, actually. Jake. Anyways, he just distracted me for a minute. And so the question becomes, down here, if I walk by and tap you on the shoulder, all right, versus I walk by and punch you in the throat or in the face, all right, how do we distinguished. If a pain cell is a pain cell, how do we distinguish between a lot of pain and a little bit of pain? Well, the number or the reason is that it's the number of neurons that fire. All right, all the pain neurons will fire. All right, but the bigger the pain, the more neurons is what's going to fire. And as an example, squeezing a trigger harder won't make the bullet go any faster. Whether I do the slow squeeze or pull it like this, all right, that bullet's going to go no matter what. All right, but if I've got a big wall up here and I shoot one gun, bullet hits the wall, I might make a dent. But if I've got 10,000 guns and I shoot them into the wall, all right, that wall's going to feel it. It's going to make a big difference. That wall's going to fall. Okay, so again, that's an example of the all or nothing principle. Depolarization is just what I was talking about with the ions. It's the initial movement of the action potential where the action passes from resting potential in the cell body into the action potential. All right, so this is essentially when it goes from resting to firing. And here's an example of that. I'll let that run a few times. The signal comes in, action potential happens, depolarization happens, and it shoots it down into the axon terminal. Hope you understand. Okay. Watch it one more time. All right. After the cell fires, there is a period known as a refractory period. And essentially, that is the cell recharging itself. Most of you, since we all have cell phones now that have cameras on them, most of y'all have probably never used or made a disposable camera. Disposable cameras, you had to, when you bought one, in order to use the flash, you had to charge it. So there, you can see this little button right here. You had to push to charge. So you push it, and it would charge, I guess, the capacitor or whatever, so that it would be ready to flash. And then if you needed a flash, you could take the picture. All right. Well, once that was taken, you couldn't take another one with the flash. You had to recharge it. Okay. And the refractory period is essentially a recharging period. Each action potential is followed by a brief recharging period known as refractory. After the refractory period, the neuron is capable of another action potential. Again, after the refractory period, it's been essentially recharged. It can go. It'll be interesting to find out if any of you have ever actually used a disposable camera. So. Yeah. All right. Remember I talked about how the cells connected? Well, in reality, they actually don't. They're not really connected. All right. There's actually a minuscule gap between each cell, and it is called the synapse. And what this does, this keeps the charge from jumping from cell to cell. Because again, a lot of times it doesn't need to. There are sometimes the cells are gonna decide, you know what? No, it does not need to be sent any further, and it stops. All right. Well, this is one of the ways that it stops. All right. The signal will enter through the axon terminal and then into the, this is the axon terminal here. All right. Well, this gap between the axon terminal and this dendrite is called the synapse. To pass across the synaptic gap or the synaptic cleft, all right, and that's what this area is. This whole area, I'm sorry, yeah. The synaptic gap or synaptic cleft, an electrical message must go through a change in the terminal buttons. All right, so these areas here are called the terminal buttons. This here is a terminal button. This is in the axon terminal. So again, I guess going through the whole process, dendrite, cell body, neuron, axon terminal, and then this right here, the terminal button. And what happens, there's a change that takes place to the signal. It goes from an electrical signal to a chemical signal. And that transition is called the synaptic transmission. The electrical charge is turned into a chemical message that flows easily across the synaptic gap. All right. So electrical impulse comes in here and then hits the synaptic, what's also known as a synaptic button, which contains synaptic vesicles, which contain chemicals. And depending on the signal, different chemicals are released that can then travel across the synaptic cleft to the postsynaptic membrane, where those chemicals are then snatched up, and then the process starts all over again. Terminal buttons are small sacs called synaptic vesicles in the terminal buttons. These vesicles contain neurotransmitters, which are chemicals used in neural communication. All right, so these, depending on whatever vesicle they are, have different neurotransmitters. And those are going to be really important because those transmitters will decide which chemical or which signals get sent, which signals get blocked. And when we start dealing with drugs and things like that, we'll talk about how those interfere with what's going on in there. When the action potential reaches the vesicles, the signal, they are ruptured and the transmitters spill out. If they have the right fit, the transmitter fits into the receptacles like a key into a lock. All right, so if there are, each of these have a different chemical makeup, and the receptacles here have a different chemical makeup. Only that specific chemical will go into that specific receptacle, and that's something to keep in mind. And then you have a process called reuptake. Any transmitters that were not absorbed by the receiving cell get reabsorbed into the sending neuron, and the process starts over. So anything that doesn't get passed or received here gets sucked up right back in there. So, and here's sort of the whole process in a one picture. So we'll go through this. All right, the signal comes in through here. It's the dendrites. Hits the cell wall. Neuron says, yes, we're going to go. Goes through the action potential. Goes through the signal, then goes through the axon and the axon terminal. All right, now this right here is essentially this area blown up. So the action potential comes in here. It cannot jump this synaptic cleft as an electrical charge, so it has to be turned into a chemical. It hits these synaptic vesicles, which all contain neurotransmitters. These neurotransmitters are released, or these they release a neurotransmitter, and this gets blown up here. Here they're using different shapes to show the different ones. They're released into the synaptic gap, and the postsynaptic membrane where the receivers are. If this neurotransmitter molecule is a triangle or a circle, each one of these is different. So this is the shape of a triangle. Here's the shape of a triangle. Boom, this goes in here. So this has received the signal, and then it is sent on to the next one, like a lock and a key. Clear as mud, right? We'll talk a little bit more about this in class. And again, everything psychological is simultaneously biological. So we'll talk more about this in class. I just wanted to give you a brief sort of overview of what some of the things in the terms that we're going to talk about. You know, again, this is a real quick, sort of quick and dirty explanation, but you can kind of see, you know, you've got one neuron here, another neuron here. This is this blown up, and this is this area here blown up. It gives you hopefully the size of what's happening. So that is neural communication, and we'll spend some time on this. We'll spend some time on this in class. But anyways, hope you all are having a good evening, and we'll see you tomorrow. Peace.